Hello, I'm Hyung Kun Lee of Yonsei University's ophthalmology class. The host introduced the title, so I will not read it separately, and I'll start my presentation. As you know well, it would not be an exaggeration to say that it is the future medical biomarker market. I think you will hear and live countless times with the word precision medicine in the next 10 years or more. Since precision medicine actually begins with acquiring and analyzing biomarkers, I think developing and discovering biomarkers will probably be very important. As I said, I think we are at the boundary between the era with and without precision medicine, and I think we will live in an era where we can give each patient benefits through it. To get biomarker, you eventually need biomarkers biopsies, most of which seem to be slowly moving to liquid biopsies. Whether you get a dielectric material, protein, or other cells from classical biopsy, but currently in fact liquid biopsies, is moving to liquid biopsy. Liquid biopsy is the process of collectively obtaining markers for a certain disease from blood, saliva, other types of saliva, sweat, or tears, and extracting important markers from them. I'm sure you've seen it countless times. Compared to standard biopsies, liquid biopsies are extremely fast, minimal pain risk, and minimally invaluable. For example, if you have cancer and you can get it through liquid biopsies through your blood, it is the most ideal way to diagnose it without having to collect tissue. I am interested in proteomics and have been conducting research related to this with my disciple, Professor Yongwoo Ji, for a long time. Our group has been developing so-called aqueous fluid and tears markers for each disease through fluid markers in the eyes and blood markers. This video is about recruiting tears, and if you look at the video, we collect tears using polyester rod, and in fact, we are carrying out big national projects in collaboration with various institutions as well as eye diseases. That's why we're working on finding neurodegenerative markers such as Alzheimer's disease and Parkinson's, and we're doing quite well. Now you can see that the fluid coming out of the tear glands and showing you collection things through polyester rod. So we got a lot of biomarkers compared to other institutions, and through these biomarkers, we've identified that dry eye is an immune disease. That is, dry eye, not a show brain, is also an immune disease, and we checked the center of markers is immune inflammatory response. Furthermore, we saw how it changes when used with the acronym cyclosporine A, which is most commonly used for dry eye treatment, or DQS, made by a company in Japan, and confirmed that proteins change differently, as you can see. As you can see, when we used cyclosporine A, immune inflammatory pathway like MAP kinase was mainly deal with, and because decafosol is a non-immune formulation, we have confirmed that other types of pathway are more active when using these drugs. We extracted with markers that showed common treatment effects of these two drugs, obtained final biomarkers through markers, and we almost achieved results. We are developing markers that can predict Alzheimer's disease in advance using tears, such as next aqueous fluid. Alzheimer's disease is actually very difficult to diagnose, and one of the difficult reasons is that obtains the cerebrospinal fluid is very invasive, and blood is called a BBB, so it is difficult to diagnose because markers from brain do not fully melt into the blood. So what we're paying attention to is the commonality of the brain and eyes. Through this commonality, it was said that we could find markers that could be diagnosed with brain lesions in our eyes, and these thoughts for a very long time ago. In other words, the papers that are an extension of CNS have been talking about for a long time. In fact, the cranial nerve number 2 is said to be CNS. So, based on our cohort, through substances obtained from the ice, each detail played several roles and used sensors to diagnose Alzheimer's disease, and it has been working for a long time, so it is almost completed now. So I'm showing you separate things with markers, and we are working on getting some important markers from them with combination, and getting them with the equation of markers. Liquid biopsy for the pancreatic cancer is also underway, and because it is a difficult cancer to diagnose, single-cell RNA sequencing and its qPCR method have been used to find what is obtained from certain immune cells for quite a long time, and since 2016. It has been almost completed the extraction from screening marker to candidate marker, lead marker, and in fact this technology was transferred. Therefore, high-risk groups are drawing with screening markers, and candidate markers are drawn and shown. Aqueous fluid is a fluid behind the cornea. Tears flow down in front of the eyes, and aqueous fluid is a fluid that circulates throughout the eyes, so it has a lot of important information in the eyes. The most important cells in the eye cornea are corneal endothelial cells, which are honeycomb-shaped cells, that is, marked C cells, which are only 1 by 10 to 1 by 20 thickness in the entire cornea layer, but actually represent the function of the entire cornea. When you get sick, there are holes and many changes, as you can see. But there has been no way to know before these changes occur. When you get sick, there are a variety of diseases that can almost lead to blindness. As I told you, the corneal endothelial cells examination is a brief explanation, but it's quite inaccurate because it's like sampling and diagnosing something as big as a desk in a playground. In addition, there was a disadvantage that it was diagnosed only when the disease progressed, and we tried to discover biomarkers using the omics method that we have always been doing. So, we analyzed with normal control and each protein in the presence of a certain disease and extracted with biomarkers. Cataract surgery is being operated, and aqueous fluid is obtained from patients with this disease. We're showing you how to get a little bit of fluid using the 26-gauge review, and we've got a little fluid in the beginning stage and collected 1,200 to 1,300 patient data, which was probably a month ago. Through this, protein analysis was conducted and markers were used to extract markers from the so-called development stage. So we got a lot more proteins than the existing ones in aqueous fluid. And if you look at that in these proteins, we separated them with proteins that are expressed only in certain diseases, analyzed, related them, so-called not development tested with markered but in validation stage. We were trying validation for patients, and it can be confirmed that some important proteins are involved. I think these will be very important for patients to detect corneal endothelial cells with prognosis or problems in patients who appear to have abnormalities. Another thing is that I think it can be used importantly to detect how long the cornea will remain after a corneal transplant, how long it will last, and whether there is an immune response after transplantation. That's all for my presentation.